Hello, I'm Learn On Yi Teresa, and I'm excited to present the industrial manufacturing process of Penicillin G. So let's begin with the overview of our presentation. To start, let's define what penicillins are. Penicillins are a class of antibiotics characterized by a unique structure called the beta-lactam ring. This structure is crucial for their function. How do they work? Penicillins inhibit the synthesis of bacterial cell walls, ultimately leading to the death of the bacteria. They are effective against a wide range of bacteria, particularly many gram-positive and some gram-negative species. As a result, they are used to treat various infections, ranging from skin infections to pneumonia. Next, let's focus on our highlight product, the Penicillin G. This is a natural penicillin produced through the fermentation of the mold Penicillium chrysogenum. Notably, unlike some other penicillins, it is not chemically modified after the fermentation. However, it is important to note that Penicillin G is sensitive to acid, meaning it breaks down in acidic environments. This characteristic prevents it from being taken orally, so it is usually administered intravenously or intramuscularly. In addition, Penicillin G has a relatively short half-life, thus it requires frequent doses to maintain effective levels in the body. Clinically, it is used to treat various infections, such as the respiratory tract infections, sylvanus, respiratory fever, and anthrax. Now, let's explore the mechanism of the action. Earlier, we mentioned that penicillins kill bacterial cells by inhibiting the cell wall synthesis. Here's how that works on a molecular level. Bacterial cell wall consists mainly of peptidoglycan, a mesh-like structure that provides stability and prevents the cell from bursting due to the internal pressure. Peptidoglycan is formed by the long chains of sugar molecules linked together by short peptides. To facilitate this process, enzymes known as penicillin binding proteins (PBP) creating these links. So, when penicillin is present, it binds to this PBP, inhibiting their functions and preventing the proper formation of the cell wall. This weakening of the wall ultimately leads to the cell's death. Finally, let's discuss the metabolism and excretion of Penicillin G. This antibiotic is primarily excreted from the body through the kidneys, with most of it being excreted unchanged. However, some of it is metabolized in the liver, where the beta-lactam ring is broken down, resulting in the inactive metabolites. These metabolites are then eliminated through the kidneys and liver. Importantly, since one of these metabolites can cause allergic reactions, it is crucial to consider a patient's allergy history to ensure safety during the treatment, but generally, it is safe to inject into the human body when used appropriately.
The biosynthesis pathway of penicillin consists of 3 steps localized in the peroxisomes. The first is the formation of tripeptide ACV, which is formed from 3 amino acids, L-alpha-aminoadipic acid, L-cysteine, and L-valine, which are enzymatically joined together inside the fungal cell. This step is catalyzed by the enzyme ACV synthetase, encoded by the PCB-AB gene, and this tripeptide would be the initial building block for the penicillin biosynthesis. The second step is the conversion of ACV to isopenicillin N. The ACV tripeptide undergoes an oxidative ring closure to form the bicyclic core structure of penicillin called isopenicillin N. This reaction is catalyzed by isopenicillin N synthase, encoded by the PCB-C gene. The IPN contains the essential beta-lactam and thiazolidine rings characteristic of penicillins. The final step is the side chain exchange to form penicillin G, where the side chain of IPN is replaced by the phenylacetal group to produce penicillin G. This step is catalyzed by the isopenicillin N-acyltransferase, encoded by the PNDE gene. The phenylacetal side chain is derived from phenylacetic acid, which is activated inside the cell by the phenylacetal-CoA ligase. This final step occurs mainly in the peroxisomes, specialized organelles that compartmentalize penicillin biosynthesis. There are 2 genetic modification enhancements in this biosynthesis. The first is the gene duplication, where this is controlled by a gene cluster of 3 main genes, the PCB-AB, PCB-C, and the PNDE, which are encoding enzymes that catalyze the 3 steps. The tandem duplication of these genes would boost the penicillin production by increasing the gene dosage and pathway flux. The second is the overexpression of PAX11 gene to increase the peroxisome number, as it is a key peroxisome protein that regulates the peroxisome division and proliferation. The overexpression of this gene can lead to 2 to 2.5-fold change increase in production, as it increases the proliferation of the peroxisomes, where the last step of the penicillin biosynthesis occurs. At the same time, it also increases the surface area for transport of penicillin and precursors.
This is the production overviews of Penicillin G, which include the upstream fermentation, downstream fermentation, and quality control measurement. Let's first talk about the upstream fermentations, where we transfer raw nutrient into Penicillin G through carefully controlled microbial metabolism. There are some strict operating conditions that should be followed. During the process, an immobilized cell system is adapted. The operating temperature should be maintained between 25 plus or minus 2 degrees and the pressures keep at around 20 psi. pH has to be within 6 to 6.5 at the beginnings. The medium is constantly aerated and agitated at 1 ppm and 250 revolutions per minute. Making sure there is enough oxygen being supplied through aerations will minimize the mechanical damage to the immobilized cells. The upstream fermentation process is carried out in aerobic fed batch bioreactors in most of the cases, while sometimes they do use CSTR. Now we come to the medium composition. The upstream fermentation medium is composed of lactose as the carbon sources at a concentration of 1 to 4% weight per volume. Nitrogen sources including ammonia salt at 0.04 to 0.12% weight per volume. Estrus or constant liquors at 0.3 to 0.5% weight per volume. Phenylacetic acid precursor is included at 0.01 to 0.03%. Additional sources are added, totally approximately 1%. The nutrients are added gradually to prevent substrate inhibitions and toxicities, which would occur if nutrients or precursors are at a high concentration from the start. The phenylacetic acid is administered in the dose every 12 hours during the production phase. This process takes 7 to 9 days. During the lag phase, the fungal cells are adapted to the medium environment. No significant penicillin production is observed at this stage. In the exponential phase, rapid growth occurs as the nutrients are consumed. The stationary phase follows. At this stage, the addition of phenylacetic acid is initiated, with dose administered every 12 hours, like what we have mentioned in the last slide. Secondary metabolics begins, and penicillin production reaches peaks. A rise in pH is observed, attributed to the release of ammonia. Termination occurs when the pH reaches 7.8 to 8, at which point production is ceased. After 7 to 9 days upstream fermentation, we are now proceeding to downstream purification.
Penicillin G requires precise downstream processing to ensure high purity and yield. I will be introducing the purification methods of this antibiotic. After the upstream process, the medium is first filtered to separate the medium from the mushroom mycelial fermentation broth using a rotary filter. The primary fermented broth is then cooled to 0 to 2 degrees Celsius to stabilize penicillin. Sulfuric acid is then added to drop the pH to 2.0 to 2.5. At this low pH, penicillin remains protonated, making it soluble in organic solvents like N-butyl acetate. The mixture is centrifuged to separate the organic phase containing penicillin from water-soluble impurities like sugars, salt, and proteins. This step removes around 80% of impurities. We then transfer penicillin back to aqueous phase and shift the pH to 6.8 to 8.0 using a phosphate or carbonate buffer. Now penicillin becomes deprotonated and water-soluble, leaving organic residues behind. Note that a neutral to slightly alkaline pH prevents degradation and allows further purification. During secondary fermentation, the strain consumes nutrients, releasing acids that keep pH at 6 to 7. Once glucose is depleted, acids are metabolized, raising pH above 8. At this point, penicillin production stops because key enzymes deactivate. Now we re-extract penicillin for further harvesting. The purified solution is concentrated and ethanol is added to precipitate penicillin as sodium or potassium salt crystals. These are vacuum filtered, washed, and dried to produce the final powder. For a greener approach, some adopt the method of amberlite XAD resins. Penicillin binds via hydrophobic interactions thanks to its phenyl group, while impurities like sugars flow through. The conditions for resin purification is that the flow rate should be between 1 to 2 bed volumes per hour. The rate is slow for maximum binding. The required temperature falls between 4 to 10 degrees Celsius to prevent degradation, while the pH is kept at 6.5 to 7.5 to ensure optimal stability. After washing the resin to remove impurities, penicillin is released using organic solvent such as ethanol water. The eluate is concentrated, crystallized with ethanol, and dried, just like in solvent extraction.
The two key methods for isolating penicillin G: solvent extraction and hydrophobic adsorption. Solvent extraction is a traditional approach, but is complex and costly. It involves multiple steps: pH adjustment, cooling, and centrifugal separation. It requires recycling flammable solvents like n-butyl acetate. The acidic conditions risk degrading penicillin, and residual solvents may linger, demanding extra purification. In contrast, hydrophobic adsorption uses resins like amberlite XAD. This method is simpler. Penicillin binds to the resin at one pH and releases at another, skipping harsh solvents. It's greener, cheaper long-term, and preserves penicillin stability by avoiding low pH. The result is higher yields and fewer impurities. Quality control in penicillin production ensures the antibiotic's safety, efficacy, and consistency through rigorous testing and protocols. Regular sampling using HPLC precisely quantifies penicillin while detecting degradation products like penicilloic acid, complemented by TLC for rapid impurity screening. Bioassays confirm the antibiotic's biological activity and stability, verifying its ability to inhibit bacterial growth. Additionally, manufacturers adhere to GMP and cGMP standards, implementing aseptic filling and packaging to prevent contamination. Together, these measures, from analytical testing to sterile handling, guarantee that every batch meets stringent regulatory and therapeutic standards, delivering a reliable product for patient use. This is the end of our presentation. Thanks for watching.